Great. Next group is model selection for accurate tumor diagnosis.
Hello, I'm Xiaoxu.
Oh, hello, I'm Lichao from Material Science Engineering.
So in this project, we developed some tools to help clinicians to select correct models for tumor diagnosis. Next, we will use a demo to show how to communicate with the AI. So now I'm a clinician. So in the morning, I want to do I need to do some surgery and to diagnose if the patient has tumors or not. So in the morning, I want to fresh my mind about how to do this experiment. So I will ask AI, please describe the clinical procedures of positron emission tomography. Then it tells me that this technique involves some nuclear medicine technology. So we will ask our patient to remain starvation for night. So patients are very starved. Then we will label glucose molecules with an isotope. Then we will inject these glucose molecules into the patient's blood. Then those labeled glucose molecules will accumulate in those regions which require high energy. So next, I will show. Now we inject the labeled glucose molecule into the bloodstream. Then we will take images. So what image we will get? And also, I will want to observe what's the FDG concentration in the patient's plasma. And we can see that the dots are the experimental data. Then it will linear interpolate this data and it tell us what's the plasma FDG concentration at different time points. Now we take images at different time points. So it will, and suppose this image is what we get from clinically. In this image, we can see that some regions we can observe some abnormally bright regions. It means those regions it has a lot of reactivity. It means a lot of FDG accumulates there, and those regions are perhaps tumors. So I want to analyze the cellular metabolism there. So next, I will ask AI what models I should choose to analyze the cell metabolism at those regions. Then it will tell us that 3 models to choose. One is the 2-tissue compound model, which is the simplest one and it's the most popular one. But it has some limitations. So a more general model is called the 3-tissue compound model. And the most comprehensive one is the diffusion incorporative simulation. It means we consider the diffusion of labeled glucose molecules in the tissue, and it will be and the computational cost will be expensive for that. So next, we want to ask, what if we input the parameters of patients? So what models we can choose? Now I input the physiological parameters of the patients, and also I tell AI I will take images in 1/over after the injection. Then it will analyze the criteria that we defined. It finds that the lambda2 is 10, it's larger than 1, and also it's based on the value of lambda3. So it tells us, oh, we should use the diffusion incorporative simulation, although it's computationally expensive. Now we know we have this model to consider the cellular metabolism. Usually we have many data of the animal model. So I want to compare our results with the results from animal models. But for rice, the physiological environment is different. So I tell AI that if I consider a mouse which has a smaller intercapillary distance, then it tells me that in this case I can use a 3-tissue compound model. And sometimes we don't have certain information in the animal models. So I tell the AI that what if there's one parameter is uncertain? So which model I can use at different. So it will tell us in different scenarios which model I should use. And it will analyze the lambda1, lambda3, and 2, these 3 parameters at different cases. Hope it works. Yeah, then it tell us that if the K1 is means the cellular metabolism. If the cellular metabolism is small value, then it tell us the value of lambda2 and lambda3. And in that case, we can use the 2-tissue compound model. And for high cellular metabolism, we should use a diffusion incorporative simulation. So at last, I will ask AI if I consider a region, for example, in patients that different organs, we have different vessel density. So in some region, we may have a dense vascular architecture, and in other regions, it may be contain loose vascular architecture. So I ask him, if I consider a region which contains a very low number of vascular architecture, what images that I'm likely to see? And we can see that it look like this. And I want to ask, what if I consider a dense vascular architecture? What's a typical example? So in this prompt, I neglect the large number of vessels. So, but I guess it can figure out what I ask. Yeah, perhaps let's ask him, is there a typical example of the image? Typical image? Yeah, so I guess through this Q&A, and I'm now as a clinician, which is not familiar with mathematics, now I can ask AI to help me analyze which model I should use for different physiological environment and for different organs. And also AI can tell me if I compare different animals and human and mouse model, so which model I can consider. Yeah, thank you.
Because it seems like the function just only give you. Yeah.
Here I just we define some typical images. Yeah, in those scenarios, and for we can also generate it using simulations, but it will take a longer time. So we don't have to start the prompt.
So like here, you already provide these images.
Yes, yeah, it's the typical example. So for different vessel densities, and we can also define the simulation tool in the MCP, but it will take a longer time.
I mean, simulation itself is very long.
I'm not familiar with this field. So is this scenario like you describe? You have a surgeon person.
Yeah.
Like, is this like a situation of research, or am I like doctor?
Yeah. Yeah, in clinic, we will inject, we will first label glucose molecules with an isotope. So this molecule will look like glucose and also be radioactive. So we inject them into the patient bloodstream, and they will accumulate in those regions which require high energy, because some cancer cells will absorb a lot of glucose molecules. So in the image, we will see some region having abnormally high radioactivity. Then we will analyze the cellular metabolism there, because the cellular metabolism can tell us if those are tumors. If yes, how severe it is. Then typically clinician will just use the two-component model. But recently we find the physiological environment and the vessel structure will also affect the pixel intensity that we observe in medical image. So now we encode those criteria in this tool.
On the model selection part. So is that done by the LLM itself, or the MCP server have a function that's actually doing that?
Yeah, I define function because recently we proposed some dimensionless parameters to incorporate the physiological environment and vessel architecture into those parameters to determine which model we should use.
What are the inputs to that?
Input would be the physiological parameter, for example, the diffusivity and the intercapillary distance, the patient's weight and height, those parameters.
So the LLM is like
Yeah, yes, and also I find it will convert the units very smartly. Yeah, because for clinician we usually don't understand any mathematics, and also we will use our specific units in the parameters.
Thank you.
Thank you.